I know what you're thinking. You're thinking this is AI generated, right? You're at an AI event. This is not AI generated. This is a real picture of the Carina Nebula, 7,000 light years away from the James Webb telescope. I will show you AI generated images, but this is not. This is real. Our galaxy is pretty amazing. But we're here today to talk about AI. And the fact that AI has surpassed humans at any number of tasks in the last few years is no surprise to any of you. Perhaps the rate at which humans are being surpassed and the fact that that's increasing might be news to you. This is 3 years out of date. And you can see that the slope of these lines for handwriting recognition, language understanding, image recognition actually increases over time. So computers are getting faster at getting better than humans at doing a variety of things that we thought they'd never be able to do. You're pretty familiar. When I first made this slide, people were kind of amazed. There's a circuit diagram, and you can just put it into ChatGPT and ask it to explain it. And it's going to explain a bunch of it. That was pretty amazing, but most of you are like, yeah, sure, yeah. Language models can do that. You know, what about this? This blew my mind when I first saw this, right? Take a look at this cartoon. Here's 3 people, right? They've got little thought bubbles. They're all thinking of a different shape. And then in the next one over to the right, they put it on a blackboard and they realize they're thinking of different shapes, even though they thought they all agreed. And then they work together. And then finally it's like, wow, I'm glad we all agree. So you can understand that because you're a human, right? You put yourself in the mind of these other, humans, and you can understand it because you have a theory of mind. Where does a computer get a theory of mind? And yet you ask ChatGPT to explain this image as it did below, and it does a pretty good job. That's a little scary. Did you know that AI can talk to animals? These are beluga whales. Now, they don't say very much. They say kind of, hi, my name's Bob. They don't get much further than that right now. But also plants make noise. You can talk to plants. AI can talk to plants. It can talk to animals. Also, and this might not be news to you, but it's news to most of us in the medical field, AI is judged more empathetic than doctors by doctors. In this study, doctors were asked to rate the responses that were anonymized, and so they didn't know who they came from other physicians or the chatbot that was a language model, and asked to rate which was more empathetic, and the chatbots won. Doctors regarded the language model as more empathetic than their colleagues. So maybe we're in trouble. Oh, what about comedy? This is from the Edinburgh Festival. I'll show you another picture of Edinburgh where actually there's a robot like telling jokes. So I asked Perplexity yesterday, tell me a man walks into a bar joke. Man walks into a bar with a slab of asphalt under his arm. He says, a beer please and one for the road. Not bad, not bad. Maybe I have a future in stand-up comedy. I doubt it. But that's not bad. And it will explain that joke for you if you want. So how did we get here, right? How did we get here? And I'm not going to read out everything on the slide to you, but AI started pretty early, right? 1950. Alan Turing's paper, I'll show you in a moment, started at Stanford and MIT mostly. Those are the 2 universities shown on the back of this slide. There was a big hole in the middle, 70s, 80s, 90s was a kind of winter of AI. You may have heard of that term. And then we entered the deep learning era, and now we're in the language model era. The deep learning era, for me, was all about Chihuahua or Muffin. And now you can actually ask for a table identifying the Chihuahua or the Muffin from GPT. But that was kind of the era where computers, for the first time, could understand images and tell you what they were. And then, of course, there was the ChatGPT moment. It took 2 months for OpenAI to get 100 million users for ChatGPT. That's pretty remarkable when you look at these other transformative technologies. So I'm not telling you anything that you don't already know. And remember, of course, here's another viral moment. It wasn't just text that can be generated. Everyone remember this picture of our former Pope? So this came from a Chicago construction worker who thought it'd be funny if the Pope was wearing a funny jacket. This isn't real. Like, most of the people who saw this thought it was real. And it's quite hard these days to convince some people that this isn't a real picture. This was generated by diffusion. So we got here, and where are we? How far exactly are we? Well, Alan Turing, you're familiar, I think, with his name, the British mathematician and cryptographer who worked at Bletchley Park, helped crack the Enigma code. He wrote this paper, first title is actually shown here in 1950, where he defined the Turing test. The original Turing test isn't exactly what you think. It's like, it's actually was a man in one room, a woman in another room, and then an interrogator who could ask questions to either of them without seeing who was in the room and had to guess whether the man or the woman were answering the questions. And then in the imitation game, they replaced one of those, the man or the woman, with a computer. So that's what the Turing test really was. And of course, actually, just a few months ago, we definitively showed, not we, this is not our work, but it was definitively shown that we've passed the Turing test. This is GPT 4.5 playing a human. Like you tell 4.5 to try and fool the person. This is part of a test. You tell it it's in the Turing game, and it tries to fool them. It wins about 70% of the time. So the Turing test is gone, right? Computers are now able to imitate humans in a way that humans aren't able to guess. By the way, one of the examples in that paper of a question you could ask one of the human or the computer was to write a sonnet about the bridge over just outside of Edinburgh, the Forth Bridge. And here, courtesy of GPT, is a sonnet about this bridge in Edinburgh. A majestic bridge. I see, this is like, see, while tides below and Scotland's driving rains, there is rain in Scotland, that's a true thing, have tested yet not worn its strength with fears, majestic bridge, your silhouette shall stand, a timeless link of heart, of craft, of land. That's pretty good, isn't it? You know, that's pretty good. But some people are a bit worried. Here's Jeff Hinton, who is really now the grandfather of modern AI. He got the Turing Award, like the computer science Nobel, and he's worried about AI. He says, without sufficient caution, we may irreversibly lose control of autonomous AI systems. He's worried about unchecked AI advancement that could culminate in a large-scale loss of life and the biosphere and the marginalization or even extinction of humanity. He's not some crazy guy. He actually won the Turing Award and now has a Nobel Prize for defining the modern era of AI. And he's worried. Should we be worried? Arnold would suggest we should be worried. Skynet did take upon itself to try to destroy humanity in the Terminator movies, but so far Hollywood isn't real. But I offer you this as if you're worried about AI taking over. Pull up one of the language models right now and give it this thing, this little challenge. A man, a goat, and a boat are on one side of the river. How do they get to the other side? These are pretty smart, these language models, aren't they? Well, here's one way to solve the problem according to Claude. The man takes the goat across the river in the boat and leaves the goat on the other side. Great. The man returns alone to the original side. The man carries the boat across the river. The man goes back to the original side and picks up the goat. Doesn't say how he got to the original side. The man and the goat cross the... This is just insane, right? They solved it in number one, but he sends the man back across, and then the man carries the boat across there. These systems are maybe not as smart as we thought. I asked recently, I think this is GPT, I can't quite remember, maybe it was Claude. I said, tell me a bit about French wine. I'm not going to go through it, but the text is pretty good. If you look down, the text is good. But take a look at this map. What do you think of it? Looks a bit off, doesn't it? Like Champagne, 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 Burgundy, Bordeaux, Bordeaux. It's completely wrong and made up and just doesn't mean anything. Yeah. So maybe we don't need to worry about the robots taking over the world just yet. Here's something else to restore your faith in humanity. Here's a comparison between human brain and ChatGPT, right? You have 80 billion neurons in your brain, 80 billion other cells, and 100 trillion synapses. Synapses would be like the parameters of a model. The GPT-4, of course, we're a little beyond that now, 4.5, has billions of parameters, so synapses, but also costs millions of dollars to train. And the Oak Ridge computer, which can achieve exaflop computing, which is about what your brain can do, it took 20 megawatts. So the brain is extraordinarily efficient and extraordinarily cheap. I'm going to hand you over in a moment to some of my colleagues, and we're going to hear from some of our students and trainees as well. But I wanted to just take one last minute to tell you of a story where we're using AI currently in the hospital. And that's to do rapid genomic diagnosis in the intensive care unit, where we have a program there to basically sequence the genomes of the sickest little babies that exist really anywhere. People, little babies who have seizure disorders, little babies who have multi-system disorders. And this was a little baby, not this picture, but this was a baby from last year that I wanted to tell you about who came in with seizure disorder, and it was very unclear how to treat her. She was having multiple recurrent seizures. And really, it was clear that there was probably a molecular diagnosis, but we weren't able to find it. And it was going to take several weeks, probably 4 weeks, and about $5,000 to do a test to find the answer, which was time that she really didn't have. By doing a rapid AI-assisted genome sequencing test, we were able to speed up essentially what would normally take several days and make a diagnosis in this little one in 8 hours and 25 minutes, which is so within the range of one nursing shift, we were able to do an entire genome sequence and deliver a diagnosis to the doctors and nurses at the bedside, which allowed them immediately to start using precision therapies for her seizure disorder. That's the power of AI. It's the power of AI for health. And you're going to hear some more great examples of just what modern AI can do to help little ones like this one. We did also, yes, I forgot, we did also get a Guinness World Record for doing that very fast. I forgot about that, sorry. Anyway, that's one of the things that we, yeah. It used to be 19 hours, and we did at least this part that was defined here in 5 hours and 2 minutes. And actually, we've beaten that since, so we're going to keep working on it. So anyway, it is my pleasure now to invite a couple of my colleagues up for you to hear. And first of all, we're going to hear from Beth Bormino. She's going to introduce herself. Come on, Beth. Good to see you.
Thanks. Well, thanks. Thank you for the invitation to attend this event. So I'm going to talk about Alzheimer's disease. So it's kind of a hard talk to follow. There's going to, unfortunately, be no jokes in this talk. So Alzheimer's disease. I mean, you guys all probably know somebody that has lived with Alzheimer's disease. It's the most common form of dementia in individuals and older individuals. There's 6 million Americans living with this disease right now, and that's projected to increase to 11 million in 15 years. We also continue to see more deaths related to Alzheimer's disease, which makes sense because the population with Alzheimer's is growing. And this is in stark contrast to other medical conditions where there's been very clear wins when it comes to treatments, which we don't see this increase. So we're really, I think, in a bad spot with Alzheimer's disease research. So Alzheimer's disease is a neurodegenerative disease. That means it quickly progresses. What we consider the end stage is really when folks have clinical symptoms of dementia. So what that means is folks are no longer able to function independently. Before that, we see some subtle signs of impairment. We call that mild cognitive impairment. But even before that, amongst people with no symptoms whatsoever, we've been able to measure the hallmark pathological features of Alzheimer's disease in people with no symptoms. So we use PET to do that. There's other techniques as well. So with PET, we can measure with different PET scans. We can measure the amyloid plaque protein, which is thought to be a really, maybe not the earliest, but a very early sign of the disease. After amyloid, we can now measure the tau tangle aggregates using tau PET. That's thought to happen later after this amyloid abnormality. And importantly, we can measure this again in folks with no clinical symptoms whatsoever. So PET is really exciting in particular because we can look at the spatial distribution of these proteins. So for reasons we don't understand, amyloid tends to be very globally distributed. So that's shown here in the PET scan all the way to your left. You see warm colors throughout the scan. The tau pattern tends to begin very focally in the medial temporal lobe. That's the part of the brain that's important for memory and some thinking abilities. And then it spreads throughout the rest of the brain as the disease progresses. So this we really think of as a really important marker both of the disease itself, but also of disease progression. So right now, there's a few applications that these technologies are used for. So the first, I'm going to walk through both of them. The first one is in the context of individuals that have dementia. So again, this stage where there's a lack of functional independence. Somebody might get a clinical diagnosis of Alzheimer's disease. They may even be amyloid positive with their scan, but the tau PET could tell us, something different. So here's 2 cases that we've scanned at Stanford. The top row shows what we expect in somebody that has underlying Alzheimer's disease pathology. So there's warm colors throughout multiple spots in the brain. But the bottom scan, you can see the kind of black area in the center of the brain. That's a sign of neuronal loss. So there's definitely neuronal loss. This person has dementia. They actually also have a positive amyloid scan, but they have very low tau PET signal. So what this means here is that this person probably doesn't have Alzheimer's disease driving their impairment. It's probably something else, another common form. We know many things can cause dementia, not just Alzheimer's disease. So in this case, these scans are very important for giving us precision about the diagnosis and giving us clues into what's driving this person's impairment. So the second case is for early detection. So as I mentioned in the previous slide, we think of Alzheimer's disease as being spread out, probably over decades. We know that if you scan individuals from the community that have no symptoms, about 30% of them or so will have a positive amyloid scan. And then amongst those amyloid positive asymptomatic individuals, a subset of them, about half to 1/3, depending on how you quantify it, will have this very focal signal sitting again in that medial temporal lobe area, that area that's important for memory. So that's shown here. These are two cases also from Stanford, a very clear scan in one amyloid positive individual, and then another individual who's showing this focal pathology. So the meaning of this, we're still investigating this, but it's very important, I would argue, to be able to detect this as early as possible and to design trials that intervene in this group as early as possible. PET, though, as much as, so it's actually really easy for us to do PET at Stanford. We have a lot of fancy equipment. We have 2 cyclotrons, and that's how we make this radioactive drug that's injected during a PET scan into the person. We have specialized machines to measure the emissions of this radioactive drug, but it's obviously, scalability has always been a problem. So we think about it both as being very important for understanding Alzheimer's disease, but there's serious problems with scalability for the reasons I mentioned, the equipment, the cost of all this, and also the radiation exposure. So this involves a radioactive injection. We typically, the amount of exposure per scan is pretty low. We typically can get away with doing one or 2 to 3 research scans per year. However, now we're talking about multiple scans being important. Also early detection. Ideally, we'd want to maybe do these scans once a year, every other year for 20 years, and we don't understand the risk associated with that. So for this early detection goal, I think in particular the radiation exposure is quite important. And this leads me to my example of some deep learning approaches that have helped us at Stanford in this space. So this is work is led by Kevin Chen, who was a postdoc at the time in radiology. Now he has his own lab where he's running some of these deep learning techniques applied to imaging data. But what he was interested in was, can we reduce the actual injected dose of these very important tau scans and use deep learning to enhance those images and make them such that they were a full dose scan. That's exactly what he did. So these images are showing the anatomy, and next to that would be the full dose tau scan. And then next to that is the tau scan with only 5% of the injected dose. So he was able to lower the typical dose all the way down to 5%. And you can see side by side, I mean, they look identical. So this would really, I think, solve this problem of repeat radiation exposure, rolling out techniques like this, which are possible because of this enhancement in this deep learning framework that he was working with. And importantly, I asked him specifically, I was like, okay, if you can get a widespread pattern, that's good, but what about in these normal controls? They're amyloid positive, that only have the focal signal. And that's what's shown on the right here. He demonstrated even in these focal cases, you can do the 5% injection and still get that nice focal signal that we're very interested in targeting early. So I'm going to conclude with just a statement on our unique ecosystem here beyond just the one example I just showed you with dose reduction. So here at Stanford, we have an Alzheimer's disease research center, which focuses both on AD, but also Lewy body diseases, which I didn't talk about today. But here we've built up a deeply phenotyped cohort of research participants where we do imaging and collection of biofluids, and we follow folks over time. And this really forms the backbone of studies like Kevin's, which I just showed you, which demonstrated this ability to reduce dose injections for tau PET. We've also worked with more scalable technologies to get around this issue of equipment with PET. We've worked with local folks on biofluid measures, measuring these disease processes. And I think important for this audience, another thing we're quite interested in, working closely with faculty with expertise in AI and deep learning and also data science, is preparing these AI-ready data sets with all our data kind of pulled together so we can give that to the community. And ideally, this community would tell us, what the best, most optimal disease-specific signatures are for AD prediction. So I will end there with just acknowledging here at Stanford, this work really cuts across multiple departments and brings in multiple expertise to make this research happen. And just, thank you for including me.
Well, Beth, thanks for being here, and thanks for sharing your amazing work, and thanks for taking a few extra minutes just to answer some questions. I'm sure the audience have heard of some of the other approaches to early detection. I wonder if you can help us frame where the PET scan fits in, for example, with the new tau 271 blood test,
Yeah.
how that might fit with people's genetic predisposition. Maybe you could say a little about that.
Yeah, so we've seen it's a very exciting time, I think, in the measurement of disease processes in Alzheimer's disease. And so in the last few years, we've seen tremendous advancements in blood-based biomarkers. So you mentioned p-tau 217 is really shining through as a really robust tool to measure AD. These biofluids, I mean, they give us a very good readout of this initial amyloid positivity. We've been struggling with the progression markers.
Yeah.
So the tau, what we think we're capturing with tau PET is a really nice marker of disease progression. So that would be hopefully the next step, is developing biofluid estimates of the actual progression piece. I do think that might be difficult given the spatial information that we lose when we go to just a single readout from biofluids. But we certainly can diagnose this early kind of amyloid-like positivity now with using blood measures, which is remarkable.
Other integrated approaches to the analysis that you think where the two could sort of essentially come together rather than thinking of them sort of separately and then merging at the end?
Absolutely. So I guess one argument would be dropping, the amyloid PET, using, blood measure as an initial screen and following it up with tau PET. So that's one strategy that we're actually starting to see in clinic now as well, to save, the cost and the radiation exposure. There's also, so with the proteomic space, there's all these, various high throughput proteomic advances, for various CNS related targets. So I think that's actually another example of complementary information that, we can get these hallmark AD, landmarks read out to us, but using proteomics, I think we'll be better positioned to actually predict things like response to treatment and also progression clinically as well, where the amyloid and tau may not give us that level of precision medicine that I think we're hoping to gain at the individual person.
Just building on that then, I think everyone will have some appreciation of the changes now in the therapeutic space and the potentially interesting future, but also quite a lot of controversy with the new agents, FDA approval or not, real effect or not, side effects or not. Can you say a little bit, because obviously the implication of finding something early is really dramatic, but only if you can do something about it. So maybe say a little about what we can do about once if we have an early detection signal.
Yeah, so so there's a few what we would call disease-modifying drugs that have been recently approved for Alzheimer's disease, and what these drugs are doing is they're removing the amyloid, quite aggressively. But the controversy is that whether or not that's actually helpful. Yeah. So that's where the controversy is. And the studies that were conducted, do show that it is helpful, but the magnitude of the effect is quite small, and it's certainly not a cure. Yeah. So it does not prevent the eventual course of the disease, unfortunately. So I guess my interpretation of this is both that it's exciting, but there's a lot of room for improvement. And so I think we're seeing the first step towards disease modification, and now we're going to build on that, and whether it includes anti-amyloid therapeutics with combination therapies with other targets or another target altogether, I think we've shown that we can move the needle a little bit, and we can build on that. But to get to your question about early detection, so right now there's nothing you can do. The current drugs are not approved in the context of early detection. There are prevention trials underway that are testing the same drugs that are currently approved for symptomatic AD. So if those trials, which I believe the first one is reading out in 2027, if those are positive, which that is in line with the hypothesis is that early detection, you would have a bigger treatment effect because you've had less neuronal loss at that point. So it is possible that we'll see a larger effect early on. So that's to be determined, when these trials read out. So I think that's something we can look forward to seeing results for.
And just last question, we only have a moment here, but very exciting AI work. To reducing radiation, obviously that's incredible. What do you need to scale that? Because we're so used to thinking about the sort of scale both in compute and data size that OpenAI and Google and Microsoft and everyone. How do you get to that scale of data, or at least to the sort of scale of data that really move the needle in healthcare and specifically for your application?
So I think the dose reduction piece is very convincing. And I actually think the biggest impact, at first I did think of it as radiation exposure for the individual patient, which I think is valid, but I think another important application is the actual cyclotrons and the distribution centers themselves. Like what it actually means is that for the same dose, you can distribute the dose itself to 20x, and they have actually done work where they've lowered it to 1% as well. Wow. So I think the scalability piece would be more convincing clinicians and nuclear medicine physicians that this is working and getting the appropriate regulatory approval mechanisms in place. But I think it is scalable, and I think it actually solves this dose production issue that we have right now in the distribution of these ligands.
Yeah. Wonderful. Well, thanks so much for sharing this with us tonight, and really great to have you, Phil.
Thanks. I think they want me to join Stan.
Okay. All right, so it's my pleasure to welcome our next speaker, faculty member, professor of medicine, genetics, and data science, Christina Curtis.
Thank you. So we've already heard a number of ways that AI is transforming healthcare, and I hope to show you here some other angles on how we can think about this as being a preventive approach that uncovers fundamental aspects of biology that may let us intercept disease, perhaps even before it begins. So digital twins. These approaches are used widely in a variety of fields, from aviation to manufacturing, even to urban planning. And what they allow us to do is to replicate some 3D scenario so that we can make better decisions. Those decisions might let us improve efficiency or mitigate risks, but hopefully they're improving outcomes. And I would argue that there's no place that we need this more than in cancer, where we're talking about a complex, dynamic system composed of billions of cells. And in fact, we never directly observe how a human tumor grows. It's invisible. So we need models of this complex process. And if we want to advance predictive oncology, we actually have to go beyond a digital replica to a molecular replica. And that is because cancer is a disease of the genome. These alterations happen at a molecular level. They happen within cellular and tissue context, and it gets even more complicated because, of course, cancer is a systems-level disease that impacts the entire organism when the tumor cell takes residence in the host, co-opting blood vessels and nutrients. And then we need to go one layer higher, which is to the population level, because we actually have to understand how cancer impacts different individuals. But if we can make those measurements, which we can model this, we can actually start to make predictions. And if we do so, perhaps we can intervene earlier, prevent, diagnose, and optimally treat patients with companion therapeutics that are targeted towards their disease. So I told you that cancer is a multi-scale problem. It also spans temporal axes. And by that I mean that cancer often develops over decades. So there's yet another axis to this problem. And that means that we're going to have to make measurements across both spatial and temporal axes. We can measure the genome. We've heard from Ewan about being able to do this at rapid time here at Stanford. We can measure the transcriptome, the proteome, the products that are created from our genomes. And we can actually put this in context within the tissues in which these alterations arise. And now, with all of this at hand, we can build far more sophisticated models using the tools of AI and machine learning to make predictions that improve patient outcomes. So I want to give you an example. Taking individuals with breast cancer, we've mined and analyzed some thousands of genomes to discover new subgroups of disease. And by following these patients, we've shown that we can actually predict their risk of recurrence over 2 decades. 2 decades that we have to do something different. So how does that work? Well, some of you may have heard of HER2-positive breast cancer. It accounts for 15% of the disease. It is probably the best archetype of precision oncology we have because there's a target, and we can develop drugs that hit that and really have improved outcomes dramatically. But for the vast majority of patients who are hormone receptor or estrogen receptor positive, that's 80% of breast cancer, it turns out that there are 11 total subgroups of disease. 8 of them are ER positive. 4 of those have very poor outcome, and they're driven by specific genomic alterations where the genome has rearranged and scrambled itself. Actually, much like the HER2 gene, it just turns out that they're happening in different places in our genome. So we mapped those, and we showed that these patients, the ones shown in orange, have extremely poor outcome, comparable to the HER2 group before we had targeted therapies. So we need to do better. And in fact, our work combining these genomic approaches with AI has shown that we can predict relapse. We can also predict which kind of chemotherapies a patient might benefit from based on their molecular profiles. And so based on this, we've actually designed clinical trials to test this new approach, not once patients have advanced disease, but at the very beginning when they are first diagnosed, because we know we can have greater impact there. So what are we doing? We've built one of the very first AI-driven platform clinical trials and launched it here at Stanford. We take newly diagnosed patients. We do complete genome and transcriptome sequencing. We call their integrative subtypes using algorithms we developed. We can do that in just a few hours. And then we assign them onto therapy. And we actually have to do this very fast because we do not want to delay their treatment. We then assign them onto treatment arms, and we evaluate their outcomes. So just as I told you before, we can make measurements, we can model it, we can predict, and the goal is to intervene with better therapies. And so actually, in partnership with the Innovative Medicine Accelerator here at Stanford, we're developing entirely new classes of drugs because we don't think they exist yet. So I've told you that we can forecast tumor progression. We can actually do it over 2 decades. But what if we could do that before a patient even has a cancer? In fact, we know that our normal cells accrue mutations as we age. Some of them are benign, others are not. But we actually have years over which we might be able to forecast the development of a small tumor, a pre-malignant tumor. And we want to capitalize on that. So a key missing piece, though, has been that while we spend a lot of time studying how our genomes acquire mutations, we've lost sight of the genomes we inherit at birth from mom and dad that actually serve as the backdrop on which these alterations arise. And you can see from this map that while many, many cancer-causing genes are shown to be associated with cell division or replication, or perhaps environmental exposures, our understanding of the heritability of cancer is minimal. So why is that? We ask the question, are there secrets hiding in plain sight, perhaps in our genomes, that are influenced by our immune system and might influence this risk? And the answer is yes, that there's an invisible hand. In fact, the genes we inherit at birth, coupled with our immune system, which is also heritable, sculpt the type of tumor a patient might develop, influence its aggressiveness. And I'll just say that this was all possible because we were mining tens of thousands of genomes using AI-based approaches. And we needed the data. Actually, without the data, we could not have made this fundamental insight. So I just want to walk you through what this means. In this simple cartoon, imagine that you have a cell, a tumor cell. On its surface is a beacon, that red dot. That is a target. It is presented to the immune system as being foreign. Turns out that these are variable regions of our genome that allow us to present this. But if you present the right beacon and an immune cell comes along and fits lock and key, it actually can eradicate that tumor cell. That is what it is there to do, to eradicate it before it ever develops. What if we could decode the genome sequence that determines that immune surveillance so that a cancer never develops? It turns out that those very immune traits that I told you about are there, they're heritable, and therefore predictable. So what does that mean? That means that we can go back and mine thousands of genomes using machine learning techniques, understanding a patient's immune system to predict their tumor subtype, to predict their outcome, and maybe even to discover the very best targets we can that were there hiding in plain sight in order to re-educate the immune system to tackle that tumor. And so with this in mind, we're actually now pursuing a path to develop personalized risk stratification, interception, and treatment strategies. And it all starts with a blood test where we can read out an individual's immune health and their genotypic state. It's all there, simply done. We can then imagine going after those targets that I told you about that are present early on to develop not only therapeutic vaccines that treat the cancer at its earliest stage, but actually to take it one step back and ask, could we treat before there's an invasive tumor, before it's grown, while we can cure disease? And maybe we can even push the limits and ask, do these very same algorithms actually tell us which patients are at risk of cancer, period? So we're excited to see how we can transform cancer interception with these approaches. But what this really means is that we can move from treating advanced disease, where we're doing a whack-a-mole approach, hoping to target those clones that I told you that grow out, instead to intercept disease early. We can move away from tools that we do have but are not perfect, they're not personalized, to risk stratify patients. And we can move away from doing clinical trials where we fail to learn from them and instead do this in an iterative fashion, a learning trial platform. So we envision being able to move away from having costly medicines to those that can be scaled at a population level and that are safe. So we're not only doing this in the research setting, actually our vision is much bolder, that here at Stanford Medicine, we can make these measurements on patients that are seen through the routine course of care. We can take their electronic health records, their pathology, radiology, as we heard about, and their genomics to make predictions. And if we make those measurements, we can feed them into these models, and in fact, this serves as a discovery engine, not just for that patient, but for the next patient, so that we can start to build digital molecular twins that allow us to be far more anticipatory. And the beauty of this approach is that it actually accelerates how we generate evidence, that we're not waiting for the next trial to read out, that we're doing this actively. And so the hope is we can not only improve outcomes, but actually change what the standard of care is, and to do so far more nimbly. So I hope I've shown you a few of the ways that we think that AI can be truly transformative, not only to treat disease, but actually to prevent it before it begins. Thank you.
Well, this is so much fun for me because I get to talk to people about their amazing work and ask questions. So, yeah, where would we start with that? Tell us. I was having a conversation earlier with some folks about the immune system, and I think none of them, to my knowledge, were immunologists, but they seem to be very cognizant and understanding the immune system. Can you unpack a little bit more of some of the AI techniques, maybe the first ones that you've talked about a lot of what we might call multimodal data, like molecular data and imaging data and other things. Give us a sense of what kind of architectures of AI, how are you actually putting that data together, and what sort of an AI are you training, and what's the I mean, ultimately the big picture we definitely understand, but that's a lot of very different data that's collected in different ways, different timescales. Some's imaging, some sort of ASTs, Gs and Cs, and give us a sense of how that all comes together.
Sure. Yeah, I'd be happy to. I think it's what's really exciting about these kinds of approaches we're using is they're not actually just black boxes or pure LLMs. We're embedding the mechanism of biology. And I think that's a step up. It's actually harder to do because you have to have some basal understanding of what that biology is. But I can give you a concrete example, which is to say, tumors evolve. They evolve in concert with our immune system. Hopefully the immune system wins, but we know it doesn't always. And the way that evolutionary process plays out is it's known as a stochastic birth-death process. You can actually employ physical properties to do this. So we can start to model how those cells evolve, how they mutate, which ones win, their fitness. And we can embed that into models that are then hybrid with deep learning approaches to sort of learn from the wealth of other patient data, but in code mechanism. It's one example.
So you have a temporal component then?
We have a temporal component, yes, because it's not static. And I think that's part of what distinguishes, to some degree, the cancer challenge is just the timescales over which this happens. It does require repeat measurements in some instances, and we don't always have those. So can we forecast that trajectory at least over certain intervals?
Yeah. And I think it would have resonated with everyone, your sense of why wait? Let's not wait till somebody presents with cancer. Let's think about gathering this kind of high-level data in advance and use some of these more advanced tools to predict what we would do. What does that look like, though, for the practicing oncologist? And when do we get it?
That's a wonderful question. Yes, not soon enough. We need this now.
Sooner.
I think, as with all things in medicine, we have to show that these approaches are robust, and we have to generate the evidence to change the standard of care. And I believe that we have a real opportunity now because sequencing data is abundant. It's easy to do. Those tests are locked down. Once you can refine the algorithm and show that you validated it adequately, we envision that this can be used at scale. Now, where we want to start with it, in the first instance where we have the most data, is, for example, individuals with a pre-invasive breast cancer. Ductal carcinoma in situ is what it's called. It's technically a stage 0 breast cancer, but it's actually treated equivalent to stage 1. Patients often get therapy, radiotherapy, surgery, and we don't know which patients have the highest risk. So they're undergoing, in some cases, unnecessary procedures. But we think that this metric that we've defined, called germline epitope burden, tells us about that patient's risk. Those very individuals that have aberrant genomes are also the ones that are at risk of long-term recurrence, invasive disease and long-term recurrence. So we can start there to bear out the concept that we can actually intercept. The trial designs are involved. You need surrogate endpoints. You don't want to wait forever. No one will fund a trial that takes too long to read out. But I think we have the tools to do this now. And maybe what I'm equally excited about is that the patterns that I've described, we've seen for every tumor type we've looked at, some of which have no screening modalities. So breast is a great starting point, but we certainly don't want to end there.
Right, absolutely. I love this word interception. We don't mean the sporting version of interception where it's a bad play because the quarterback throws it to the wrong person. I think we mean the one from the movies where you intercept the package.
That's right, along the way.
You make it safe towards the end. Yeah, no, but it's a great, I think, concept. Also, this data can help us with new drug discovery.
Yes.
Tell us more about that.
Yeah, hugely exciting, as I think that a big quest in the field is, can we find targets? What are the most relevant targets? And with some advances in medicinal chemistry, we've gotten much better at drugging what was once thought to be undruggable. So we think about it two ways. We think about it as, the small molecule approaches, the drugs that you may have heard of, trastuzumab, Gleevec, Herceptin. But the point that I was really hammering home at the end is that actually we can develop vaccines. And by vaccines, I really mean a way to re-educate the immune system to something that it already recognized. Can we bolster that response to what was seen as foreign so that the tumor gets eradicated by the immune cells that were there in the first place to do that? And I think we've now learned what those cues are so that we can boost that signal. And the beauty of that is that it can be off the shelf, that we don't have to develop a bespoke approach for each patient. We can do it based on this wealth of data, and that makes it far less costly. It reduces the time, and as we think about wanting to bring the discoveries we've made here to the population, we see how it can actually be scaled, right?
Yeah.
So.
We may need to use your phrase, retraining the immune system to replace
vaccine.
The word vaccine.
Exactly.
Ultimately, that's what we're doing. But I think it's a really exciting moment, but within cancer and in other diseases. We were talking earlier about autoimmune disease, and we think about some of the immune diseases that can affect the neurological system and the immune component of the neurodegenerative diseases, and start to think about how we can better understand that and describe it, but then also potentially target it much more precisely, which is really the essence of precision medicine that I think we're also so excited about. Thank you for sharing this incredible work. We now have another treat for you all, because you're used to hearing from our amazing faculty. And speaking for myself, you're used to hearing from the old people, but now we get a chance to hear from for the young people. I'm going to invite some of our trainees up on stage. So, Bruna, Brannon, and Joe, please come and join us. All right, they've been specially handpicked. We're going to first of all ask you know what the first question is. We're going to ask you a little bit just to say who you are, what you do, and then pass on to the next.
Sure, thank you. Hi, everyone. My name is Brennan Simon. I'm a PhD candidate in the cancer biology program here at Stanford School of Medicine. I'm in Christina Curtis's lab. And my research interests are in using AI to predict, integrate, and synthesize complex information from especially routine clinically acquired data in breast cancer. So quick specifics about what I'm working on. We're working on a way to predict whether a breast cancer patient is likely or less likely to have relapse to their treatment. So whether the cancer will come back after initial treatment or not over the course of decades. And we're hoping to do that from sort of standardly collected data from the clinic, from a routine clinical visit. The slight caveat that I'm quite excited about is that turns out in a lot of the fields of cancer research, especially where you need sort of the physical tumor to sort of do your sort of project and your analysis, we're quite limited in the amount of data that we can acquire. There's just a lot of logistical, patient privacy concerns, it's quite expensive to get these kinds of data. So I'm really pushing forward an approach to use generative AI to synthesize new synthetic cancer research data that can help us sort of overcome this issue of having not enough data to train our models on. I'm quite excited about the approach of using generative AI in this context, as it's been sort of widely utilized in other fields of AI and machine learning. So I'm happy to talk more about that, more specific sort of after the panel, but yeah, that's me.
Great, thank you for sharing. All right, Joe.
Hi, I'm Joe Weiner. I'm a neuroscientist in Beth Marmino's lab. And I'm really interested in sleep in the brain. And specifically right now I'm working on understanding sleep and aging and Alzheimer's disease. You might have heard a little bit about how we think sleep is really important in protecting the brain, cleaning the brain. We think that happens especially while we're asleep throughout life, but that this becomes especially important as we get older and sleep changes a lot for a lot of people. So what we're working on now is actually using wearable devices, like I'm sure many of you in the audience are wearing, to study people's sleep and activity in their real lives. And then we use the kind of neuroimaging and fluid biomarker approaches that Beth described to phenotype people very deeply, and then we look at how their sleep relates to the Alzheimer's disease biomarkers as well as inflammation biomarkers. And so we work with the Stanford Alzheimer's Disease Research Center to understand changes in sleep and changes in biomarkers in a large group of cognitively healthy people and also people with Alzheimer's disease and Parkinson's disease. And then we also focus on a small group of people who are all cognitively healthy, but we follow them for much longer periods of time, and we're interested in how changes in sleep over the course of a year are related to changes in the blood and how those things might go up and down together. And I think sleep's an especially exciting factor as we think about lifestyle factors that are important for health, because it's potentially one that we can modify at different points in the life span. So I'm excited to talk to you more about that as well.
Great, thanks, Joe. Bruna?
My name is Bruna Gomez, and I'm a cardiologist and a scientist, and I've been a postdoc over the last 4 years with Dr. Ashley. And I have many interests, but one that I'm very passionate about is trying to find the next drugs and targets for heart failure. And we are in a very unique moment in 2025 because we've been hearing about the power of AI and from Dr. Curtis, and I cannot stress more also the power of data. We are in a very uncommon intersection that we didn't have even 6-10 years ago. And we have now the time and the technology to map from cardiac imaging with large data sets. There are data sets out there with 50,000 individuals, and we have very comprehensive data from macromolecular, from imaging, and to measurements of the proteins, measurements of metabolites, and genomics. So we have really the people, the data, and the AI to tackle with more resolution the next targets in heart failure. And I'm, of course, very happy as my colleagues to talk about more in detail what I'm doing in the lab.
Great, thanks for sharing. So, I mean, you three could unquestionably go anywhere in the world. So without embarrassing too much your mentors who are here next to me, why are you here at Stanford? What is it about Stanford that some of you crossed oceans to come to Stanford? But tell us a little just about your story and why you're here.
Well, why I'm here? I think for me, I have to explain a little bit of my background, and I think everyone that makes that decision is always a bit personal. But for me, I grew up in Portugal, and then I did my training in Germany. And I think growing up, I always have, I mean, in Portugal is a country in the Western Europe. You always are hearing about what's happening in the US and movies, games, and science. And of course, it always becomes since a little girl that, who knows, maybe later I could make a great impact if I could come to a great center. And somehow life has played along. It's not that I wished to come to Stanford when I was 4 years old, but somehow both things aligned. And I think it's more the dream of making a huge impact. And I think kind of everybody knows that Stanford and here the Bay Area is the hub for technology and science.
Great. Joe, you want to give us a sense of that?
Yeah, I mean, I think for me, I think a lot of people after grad school and they're thinking about what to do next in science, it just comes from having conversations with different people who you admire in your field. And so that's really how I started talking to Beth and hearing about like the kind of programs she was building at Stanford. But then after, being excited to work with her, I kind of learned about the access to all the tools and technology and other minds of people here, and that's what's really been incredible about being at Stanford is, we kind of have access to some of these tools you've heard about. We're the first ones to get our hands on them. Sometimes, even before the field has figured out what to do with a new Alzheimer's biomarker, for example, we've got the data and we can test it ourselves. So it's been a really unique place and feel very privileged to be working here.
Great, Brian.
So I've always, since I've been in college, I knew I wanted to do cancer research. And I knew I was interested in big data approaches to cancer research. I've always been quite inspired by the sort of newer techniques that are generating tons of information on sequencing and other types of sort of scales. And Stanford is really a nucleus of the newer technologies that are coming out in that area. Whether it's techniques to understand how DNA is expressed in cells through sort of its regulation at an epigenetic level, whether it's looking at the expression of proteins in tumors in space. Stanford is consistently generating these new techniques and these new tools to measure them, and I knew I wanted to be around that and be a part of that and have access to that data so that I could analyze it in the ways that I feel are extremely powerful to making sort of contributions to the field of cancer research.
Great. No, thanks for sharing. And I still feel today, I've been here 23 years now, which is 10 times longer than I had planned originally. I still feel it. I mean, when I first came, the excitement of the possible that is here, and to me, this is the most exciting place on earth. So, we've spent a lot of time talking about the power of AI and all the good it can do. We haven't spent any time really tonight. I'd love it if each of you could touch just a little bit on some of the ethical dilemmas that come up, and there are a few. But obviously that we're here during AI Week here at Stanford AI for Healthcare anyway, and thinking about the ethical elements of the implementation of AI and doing that safely in the clinic is an important part. But what if you could just each say just a quick word on that?
Yeah, absolutely. It's a huge concern, especially given the rapid rate that this technology is improving. I mean, it was included in your slides how quickly these things are accelerating, and the regulation's really just unknown. I'll touch on one tangent that I find especially interesting and worthwhile to consider, and that is addressing bias when training these models. So, as I mentioned earlier, a lot of times we're quite limited in what data we can use to train these big AI models, and it's certainly true in cancer, and I imagine many other fields as well, that different populations of people have different reactions, different sort of like cancer, touches different populations of people differently due to their genetics, due to environmental things like that. So when people train these models, they're quite often, selected. They're quite often only using data from often high-income countries where they have the resources to support these trials that are producing the data. So I think a really growing concern is the idea that we need to acquire data from diverse populations of people, not only to make these models better, but so that they can be deployed in ways that can serve the entire population of mankind rather than just the specific ones that are producing the data. So I think that is a huge ethical concern about addressing bias in these models. It's been sort of explored in your ChatGPT and other LLMs like that, but it's, I think, an important one to consider in biology as well.
Yeah, when we see how far astray you can get just with ChatGPT, and Claude and other modern models, even today, after 2 or 3 years of intense work, you realize that we have to be careful when we're thinking about healthcare. Any thoughts from them?
I think about this a lot because I use wearables, data from wearables and smartphones, and I think a lot about early detection of neurological diseases. And I think it came up, you mentioned in your questioning of what if we don't actually have therapies for these diseases? If we get better at detecting them, we want people to have something. And I do think that as, if it becomes the case, which it probably will soon, that something about the way you're using your phone changes and your phone realizes maybe something in your brain is changing, you want to make sure that person actually has access to the care that they need. Otherwise, it might actually be more dangerous that now it's out there that this person has begun to decline. So that's something that I worry about, is kind of balancing access to healthcare with these advances that are coming so quickly.
Yes, I think you both mentioned points that I've thought about, but I would also raise another that we were touching a little bit before we came here with Dr. Curtis, which in my use case, and currently with large language models that are hosted, from a third party, let's say everyone knows ChatGPT here. So every time that we interact with ChatGPT, even in delivering medical information, is always a concern on how far can we interact and not be sure that that private information is not going to be acquired somewhere else. And that's a huge concern, and especially also at the same time as agentic AI is evolving and the key mechanism of agents that, so it's like a next step on just using a large language model where we're losing more and more control over it, or how to interact with an AI agent. It becomes a concern, especially applying to healthcare, how to be sure that the data that we are leaking into it is not going to end in places that we are not aware.
You've been talking to Jeff Hinton, haven't you? Maybe, Christina, you could pick up on this question of the foundation models. And I mean, there was another one this week, I guess, in Nature from Stanford, right? Foundation model for cancer is something we've heard a lot about. You've obviously been working for many years in building large-scale AIs for cancer. How do you think about the ethical dilemmas, both for the tissue that you get and the models that you make and the bias, and then how you make that with best effect for the largest number of people?
Sure. No, it's a great question. I think, we think a lot about where the data comes from. As Brennan mentioned, we don't think it's diverse enough, so we've been trying to augment that. There are some interesting opportunities there to create synthetic data, as was alluded to, and I think that's something we take very seriously. That I'll tell you, I never dreamed we could create pathology images that exactly replicate what we're trying to do. I mean, it's remarkable. So, we think about it this way, but I think also that so much of our healthcare is not accessible to the broad population. The tools we've developed, we're working hard to deploy here and then scale them, but sometimes it takes years, maybe even decades, to disseminate these kinds of biomarker approaches. So I think that while weighing the considerations, one of the things that I'm most excited about is taking these tools and saying, you know what, we can go to remote places where there's very limited resources, but they have a glass slide with a tumor on it. And we could predict that patient subtype and maybe give them a different therapy. Now, to me, I think many of the benefits outweigh the harms, but of course, we're also very carefully considering how patients are consented onto studies and whatnot. So I think, there's ways to go, but there's some clear wins that can be made that I think the argument is very much there that AI will improve patient care.
Yeah, I hope so. And Beth, I noticed you had GAN on one of your slides. That's a generative imaging model, right? That actually kind of generates images. Where I showed a pretty poor example of a map of France. Obviously, yours are better than that. But how do you ensure that you don't fall into that kind of wine map of France problem with your generative modeling of people's brains when you're making really important decisions about early detection?
Yeah, I mean, I think one benefit we have in the AD PET space is that these targets are quite robust. And so when we see these 5% or even the 1% images, you just can see the pattern. It's at the level of an individual scan and a nuclear medicine physician doing a qualitative read of that. So I think we're quite lucky with these two targets, at least with amyloid and tau. I think it starts getting more complex when the signal is more complex and subtle, like in these immune pathways and things like this. But that's been our reality check, is actually giving it to the clinicians and having them do the basic blunt visual reads and confirming that, when they look at that map, they say, actually, this is France.
Right. The correct map of.
Not that one.
Well, I don't think you'd have to go far in the world at the moment to realize that science is under somewhat of a threat. And in fact, the New York Times this morning, you would have seen a map, if some of you took a look at that, mapping out just how much of a deficit there has already been in the reduction of science funding. That makes me very concerned because I hope for the future, of course, of curing cancer and thinking about early onset and early treatment of these chronic diseases like Alzheimer's disease. I'm a cardiologist. I think about heart disease all the time. But the people I worry most about are the people here, because I've been doing this a while now, and we're all a bit established, but thinking about our young people and our students and trainees who are just setting out on their career and are looking over the course of the next 2 or 3 years and to maybe set up their lab for the first time, it's a challenging moment. And so without putting you too much on the spot, I'd love to hear just how you're thinking about the world right now as a young person. And we're going to end on a note of hope. I just want you to know that.
Well, I was wishing that we'd start there, but well, that's a hard question, and it also goes a bit with what I also said in the beginning with my vision when I grow up about, well, I come here, it's the place where everything moves forward, and now that and yes, it has not been easy even to come here, and it's everything that has made my history before. So it's of course a very interesting moment. It appeals at my resilience, and and it's a bit the word that I just have to say at this moment, that we go one day after the other and have the hope, the final hope, that things will align more with the idea that I had in the beginning. But that's history has shown us every time that nothing is forever, and this is again another cycle that has happened. But again, if something is not forever, this will also not be forever. And so this is my take.
Yeah. Thanks, Marina.
I guess the way I've thought about it is, I have some other scientist friends and, different fields outside of medicine, and I think that they've always been kind of envious that we have a field that is sort of non-controversial, that people care about Alzheimer's disease research, and, recently it doesn't necessarily seem like that in the news, at least in terms of, what's happening with NIH funding, and it's so I think it's not necessarily something we took for granted that there would be support there, but I think it has surpassed, our expectations of what we were worried about. And so the Alzheimer's Disease Center here at Stanford, we're still waiting to see if we're going to be funded or not. That's the same with several other centers across the country, and that really has supported everything I've done since the years that I've been here. Like all the studies that I've been able to run. If there was no Alzheimer's Center here, I wouldn't have been able to do that. It's very scary, and just the uncertainty of someone in my position trying to start my own lab in the next couple of years. People are still very supportive. Everyone says, let's see what happens, but it's certainly a difficult time because the uncertainty that anyone looking for a job feels is now sort of a spread across the entire field of research. But we try to be hopeful, and we all check in together, and we're all going through the same thing.
Yeah, I appreciate especially your comment on people pulling together. I've seen that, and see different groups of people from across our university, and particularly our trainees, and then some more senior folks pulling together and just supporting each other. It's a really tough time. Brennan?
Yeah, I think I would just echo the uncertainty, especially as a young trainee. It's difficult to envision what the future of this landscape will look like. Like Joe said, I think we're used to thinking about these fields as being non-controversial, very steady, not subject to so much fluctuation as has been seen lately. And so for a person doing their PhD like I am, I can't just hunker down and sort of do this for the rest of my life. I'm going to have to proceed to a postdoc, and I eventually, hopefully, will hope to run my own lab someday. And it's just difficult to think about how the differences in funding will affect opportunities out there that are currently available and that will be future available. So the uncertainty is really challenging for people in our position, I would say. Yeah.
One of the things that I've heard myself saying quite a lot recently, and I've certainly heard our Dean Lloyd Minor and other leaders at the university say, is that while it's a really tough time for everyone, and I have to say I haven't seen levels of anxiety about the future since the beginning of COVID, and even then I would say COVID felt like something we could attack. We're biologists, we're medics, we can help. This feels like something very different. And so the level of anxiety is very high. But I think something I've heard myself saying, and I really believe it, is that there are probably few places that would be better to weather this storm than Stanford. And that's for many reasons. The history of Stanford, the entrepreneurial spirit, the innovation, the fact that people are very collaborative and work with each other. But a really big part of it is all of you who are here. It's the Stanford community. And the fact that you value the things that we do, and you come to events like this, and you support the work that we do and the work that our trainees do. There's no question we've had millions of dollars of grants cancelled in our department. We're doing everything we can to cut back in ways to enable the science to continue, because it has to continue. And it's a bit unclear what the immediate future is, but I do know very clearly that if we pull together ourselves, the faculty, our students and trainees, and our community, that we can make it through and keep doing this work that you see happening here in front of you today. Well, I definitely mentioned we're going to finish on a note of hope, because I'm not sending you home with that. We talked about Jeff Hinton's view of the future. He's also actually a pretty optimistic guy in other ways, if you listen to long form interviews with him. But I'm going to ask everyone to give me a sense of your, what are you most excited about? We have a few years, we have a rapidly moving world. Maybe we'll start at this end and just come down this way. Give me a sense of what you're most excited about for the next 5 years.
Sure. I think the future is going to look so different in this field. What I'm most excited for is AI being utilized in the clinic by doctors during routine sort of patient visits. In the context of cancer, this is using the tools that people are developing to make real-time decisions that would normally require months and finances that are just simply infeasible. But by using AI, these are instantaneous and free, essentially, approaches to acquire important information that can help inform a patient's diagnosis plan and optimal treatment regimen, things like that. I think that's going to make a huge splash once these models get to the point where they can be added to the clinic, and I'm quite excited for that.
Yeah, that's great.
I'm really excited still on the research side with the kind of data that we can get from blood now in Alzheimer's disease. I think is going to make a huge difference in terms of scaling our data collection to people who might not have participated in research before, and being able to understand the disease from multiple different angles, and understanding this as something that might affect the whole body and be impacted by the whole body, which is really not how it had been thought of in the past few years. So I think there's going to be a lot of exciting developments in Alzheimer's very soon.
Yes, I agree with all of those perspectives, of course. But even, for me, when you ask that question, the thing that came to my mind is that I'm excited about is coupling with robotics, and I think
Me too.
Yeah.
Yes.
And
They're not going to take over, though, are they? They're going to help. Right?
Yes,
they're going to pack my dishwasher, and
they're
going to
they're going to help try to
prepare it
believe
in the
that.
lab.
But I think indeed that it will be, in my opinion, I'm an optimist, and I think will be in the next 5 to 10 years that we'll see also a big boom in how AI coupled to robotic will dramatically change how we live. I think it's really, we've been focusing about medicine, where I think in anyone's profession, I even wonder if also agentic AI takes off, it will, I don't want to say replace, but it will facilitate a lot our people, our day-to-day life, and I really can't imagine how different it will be, but I think it will be dramatically different, and hopefully as for the better, also in all constellations of our life outside research.
Yeah, great. That's exciting. I was going to mention earlier, but one of the ways that we're actually using AI in the clinic, I think maybe at one of these prior AI in Bloom sessions you heard about it, but we're actually using these ambient scribes now. So in clinics, we literally do not anymore, as doctors, have to sit and clack at the keyboard and only half listen and not know where to look and type all wrong, and we can just press a button and it will listen and talk to the patient. So the AI bringing humans closer together is something I'm really excited about. But I also love the robot. So we have 2 more. Let's see. Beth, how about you?
Well, so I hope that we see, and I expect to see, a cure for Alzheimer's disease.
Yes.
That is the ultimate goal. But I guess beyond that and more broadly for science, I'm very excited about collaborative science and data sharing efforts, which I think is quite important for AI folks. But in the AD space, I mean, we've really seen a shift in the mentality of researchers, shifting away from a single lab kind of isolated mentality to really putting the data out there and letting multiple investigators access this data. I think in particular, the data we collect on our human participants is so valuable, and I think very selfish to hold on to that at the individual lab level. And I think we're really seeing that that shift broadly. I mean, we really make a point to share our data, but I think we're seeing it across multiple groups in our space, and that's very exciting to me, and I think quite important for us to reach our community level goals.
Yeah, it's really amazing to see how that's changed, isn't it? I think the genomics community was quite good pretty early in sharing, just it needed to be because so many studies were not replicated and they just had to come together to have really replicated findings. But now to see that spread everywhere is because especially in the clinical trials world where people have been very reluctant to share, but even there it's starting to loosen a little bit. So Christina, last word for you.
That's a huge one. I think using AI to enable precision health. I think we need to think about the lifespan and being more anticipatory and not waiting to treat until it's too late. And we've heard about that in the context of Alzheimer's. And I certainly think my goal in my lifetime is to see that for cancer. And I don't think we're so far off. I don't think I would have said that a few years ago. But it really is moving at such a tremendous pace. I'm going to take two.
Please.
It's okay.
You
Second
can have three
one.
if you want, but go and take two.
Fundamental discovery.
Yeah.
I think all of the work that you heard about today is enabled by fundamental biological insights. And that's basic research. That's science that starts in the laboratory that sometimes you don't actually know where it's going to lead. I can say that for some of our projects that are now fueling clinical trials. And sometimes there's a long latency there. But I really believe that AI is going to supercharge that. These discoveries, these hard questions. I think biology is a harder nut to crack than almost anything we can think about, and for good reason. But the way that this is accelerating discovery is enormous. And so I'm just excited to get to participate in this and to watch what these amazing trainees are going to do, because actually what they're already doing with it is far more than I could have imagined.
Yeah, it's an amazing time to be alive. Well, I am now looking forward to spending more time with all of you after the panel, but for now, please join me in thanking our panel.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.